Our protein of study is a neurotoxin purified from the venom of Bungarus multicinctus, Taiwan banded krait, known as beta-bungarotoxin. Snake venom has been known to perform a variety of functions for the Taiwan branded krait, including prey digestion and immobilization. However, beta-bungarotoxin is specifically a neurotoxin which acts at presynaptic terminals and causes a loss of controlled muscular function. Throughout the video, the structure of beta-bungarotoxin will be described and depicted along with how the protein functions and affects humans in particular. We aim to show that studying the structure and function of beta-bungarotoxin enables insight into the medical relevance of this toxin and, as a result, improvements in biomedical technology. The beta-bungarotoxin protein consists of two polypeptide chains, A chain and B chain, that are cross-linked by an interchain disulfide bond. It has been determined that there are 16 isoforms of the protein, but only 8 of which have been characterized. Nonetheless, all A chains are structurally homologous to phospholipase A and are shown to be the catalytically active subunit in binding calcium and activating phospholipase activity to induce a neurotoxic effect. Meanwhile, the B chains of the different isoforms of beta-bungarotoxin differ slightly and can operate differently, showing similarities to trypsin inhibitors, toxin 1, and dendrotoxin. The A chain has 2 calcium binding sites, shown as purple spheres in this image. The red colored residues are the catalytically active amino acids that interact with the ions. Schematically, they are shown to interact with the ions as such, represented by sodium ions in this particular instance, but operating with calcium ions biologically. The A chain's catalytic mechanism is driven by histamine at amino acid residue number 48 and aspartate at AA residue number 94. The representational mechanism shows a standard phospholipase reaction. Specifically, the protein catalyzes the hydrolysis of phosphatidylcholine into acylglycerol phosphocholine and a carboxylate. This mechanism takes place as beta-bungarotoxin interacts with water and calcium to attack phosphatidylcholine and separate the molecule by division of a carbon-oxygen bond. While B chains differ structurally across isoforms, one form study acts as a trypsin inhibitor. For this isoform of beta-bungarotoxin, the trypsin inhibition is of the Kunitz family of serine protease inhibitors. The cis-aryl bond is found to be stemmed from a positive arginine residue connected to an alanine residue. This P1 site acts as a substrate for serine proteases instead of the typical biological substrate.
In determining the biological implications of this protein, investigations of the protein binding have shown that the protein binds to a single saturable class of binding sites of membranes with dissociation constant values in the range of 0.4 to 0.6 nanomolar. At the terminal regions of neurons, the protein inhibits the release of neurotransmitter at the neuromuscular junction. As a side effect, beta-bungarotoxin depolarizes mitochondrial phospholipid bilayers due to the fatty acids produced by its phospholipase A2 activity. This is shown in this image. The main function of beta-bungarotoxin slowly depolarizes the entire plasma membrane of synaptosomes. It was found that dendrotoxin partially protects against the effect of beta-bungarotoxin, suggesting a plasma membrane localization with a specific toxin binding site in intact synaptosomes. It was determined using rat brain cells that beta-bungarotoxin is highly selective to calcium ions. When measuring the release of S-acetylcholine from synaptosomes at high potassium chloride concentration, it was found that the toxin had almost no effect. After the toxin is injected by the snake, its active site is obscured, reducing hydrophobicity. This allows it to avoid binding to non-target membranes. Once reaching the presynaptic membrane, the Kunitz domain binds to the channel receptor. Substrate binding displaces trypsin 19, flipping it into the membrane, where it provides a hydrophobic anchor. Fatty acids, released upon phospholipid hydrolysis, increase the anionic composition of the membrane, enhancing beta-bungarotoxin's nonspecific electrostatic affinity. Extensive phospholipid hydrolysis interferes with neurotransmitter release, resulting in blockage of neurotransmission. This Lineweaver-Burke plot shows a dramatic decrease in choline uptake with increasing concentrations of beta-bungarotoxin.
The medical relevance of beta-bungarotoxin has been seen through various studies that test its role in biological systems. In particular, one and others examine the use of antimicrobial proteins as an alternative for antibiotics. With antibiotic resistance on the rise, this is a particularly relevant point of interest in research today. In their paper, Antibacterial and Membrane-Damaging Activities of Beta-bungarotoxin B chain, Wen and others found that snake venom can cause growth inhibition in E. coli due to an increase in membrane permeability caused by beta-bungarotoxin B chains. Even without the A subunit present, lone B chains of beta-bungarotoxin can cause membrane damage such as wrinkling, surface roughening, and membrane flabbing, shown here. This antibacterial activity is found to be dose-dependent. E. coli growth is inhibited at half its maximum rate at 8 micromolars of injected B chain. This effect is time-dependent as well. When treated with 50 micromolars of B chain, 80% of E. coli are killed after 4 hours. Wen and others were also able to specifically determine that 12 micromolars will induce 50% membrane permeability. This research has implications for future use of antimicrobial agents that circumvent current issues associated with antibacterial resistance. The beta-bungarotoxin protein exhibits a core functionality of phospholipase activity with its A chain and variable functionalities in different B chain isoforms. While only specific isoforms were detailed in this video, the study of others could potentially lead to larger discoveries in the neurotoxin field. Furthermore, beta-bungarotoxin and the study of its effects has already demonstrated possible medicinal benefits as an antimicrobial agent and could be used in a variety of other ways that have yet to be explored. Overall, research of snake venom, neurotoxins, and beta-bungarotoxin can offer key insights into structural and functional protein understanding. With successful findings in preliminary research, additional research should be pursued to continue analyzing the medical capabilities of beta-bungarotoxin.